Welcome back to the Deep Dive, where we take a look at the most recent developments in genomics and proteomics. We've got a fantastic collection of articles and research, hot off the press, ready to unpack and explore how these advances are changing the way we understand health and disease.
Yeah, it's pretty amazing the level of detail we can get into now, even down to the individual cell level.
Definitely.
Yeah.
One of the studies that really caught my eye was this one on a new technique called DSE-IC. It lets scientists actually study how DNA is folded and organized within a single cell.
It's like almost taking like a high-resolution 3D picture of DNA origami.
Oh, wow.
And it's crucial because the way DNA is folded can actually impact how our genes function. Imagine if you had a cookbook, right? And a really important recipe was like tucked away in some hard-to-reach corner.
Okay, yeah.
That's kind of what can happen with genes. If they're not easily accessible because of how the DNA is folded, they might not get expressed properly.
That's a fantastic analogy.
Yeah.
So understanding DNA folding could help us understand why some people are more susceptible to certain diseases.
Exactly. Or even why they respond differently to certain medications. It could even help pave the way for personalized medicine tailored to an individual's unique genetic makeup.
Wow, so powerful.
Yeah.
Another really interesting article I saw highlighted this computational tool called MOSCOT, which is like revolutionizing the analysis of single-cell data. Can you explain a little bit about what makes MOSCOT so special?
Moscot is kind of like, if you think about like a master chef who combines all these different culinary techniques to create this incredible multisensory dining experience.
I like that.
In the context of single-cell analysis, it lets researchers actually integrate multiple types of data from the same cell, like gene expression and protein levels all together.
So instead of just looking at like one ingredient, you can see the entire recipe and how everything interacts.
Precisely. It's essential because cellular processes are so complex and interconnected, and MOSCOT helps us understand this intricate dance of molecules within a single cell to get a much richer understanding of how cells function.
Okay, so from single-cell analysis, let's shift gears a little bit and talk about another really complex system, the gut microbiome. One of the studies we have really delves into the constant battle between our immune system and the trillions of microbes that reside in our gut.
Yeah, it's a constant balancing act, right? Our immune system needs to be vigilant to keep those harmful microbes in check, but it also has to tolerate the beneficial ones that are essential for our health.
Definitely a delicate balance. And this research used RNA sequencing to track the interactions between our immune cells and the gut microbes. Can you talk a little bit more about how this technique works and what insights it provides?
Sure. So RNA sequencing allows us to see which genes are being switched on or off, both in our cells and in the microbes. It's like we're eavesdropping on their conversations, understanding how they communicate and influence each other. This gives us really valuable clues about how our immune system responds to different types of microbes.
Wow. So deciphering a secret language, understanding the crosstalk between our bodies and this vast community of microorganisms that live within us.
Exactly. And what's really fascinating is these microbial conversations can impact various aspects of our health: digestion, metabolism, immunity, even brain function.
Really highlighting the interconnectedness
Mm.
of all the different systems within our bodies
Mhm.
and the importance of maintaining that healthy balance in our gut microbiome.
Absolutely. Sometimes those conversations can take unexpected turns. There's this other study, it highlighted a fascinating link between our immune system, blood clotting, and zinc
Oh,
ions.
that's interesting. I never thought those three were connected. What did they find?
So the study suggests that IgG antibodies, those are like key players in our immune response. Fibrinogen, a protein involved in blood clotting, and zinc ions, they're essential for lots of different cellular functions. It seems like they're all interacting.
So how are those seemingly unrelated processes linked? That's so strange.
Well, the research suggests that IgG antibodies and fibrinogen, they might actually interact with each other, and then both of those might interact with zinc ions. So it really raises questions about how these interactions could affect both our immune response and the blood clotting process.
Huh. So could this have implications for like treating diseases related to either the immune system or blood clotting?
Oh, absolutely.
Yeah.
Like understanding how zinc ions influence the interaction between IgG antibodies and fibrinogen could lead to new treatments for autoimmune disorders or even bleeding disorders.
Wow. It really shows you how everything is connected.
Yeah.
Okay, so moving on from the immune system, let's talk about the exciting field of proteomics. The study of the proteins that our genes code for.
Yeah, proteins are the workhorses of our cells, carrying out a vast array of functions that are essential for life. Just like genomics has revolutionized our understanding of DNA, proteomics is now revealing this intricate world of proteins.
And one of the articles we have actually talks about this groundbreaking new tool called Olink Reveal. It's making targeted proteomics more accessible to researchers. Can you explain what makes Olink Reveal so revolutionary?
So traditional proteomics methods, they can be really expensive and complex. They often require specialized equipment and a lot of expertise. But Olink Reveal actually simplifies the process significantly, making it more accessible to a wider range of researchers, especially those who are maybe in smaller labs or have limited resources.
So it's kind of like a more streamlined, efficient way to study proteins.
Exactly.
What are the specific advantages of using Olink Reveal?
So Olink Reveal focuses on analyzing a specific set of around 1,000 proteins that are known to be really relevant to human health and disease.
Okay.
It's kind of like, exome sequencing in genomics, where researchers focus on the protein coding regions of the genome. It's the most informative part for understanding disease.
That's a great analogy.
Yeah. By focusing on just the most relevant proteins, researchers can save a lot of time and resources and still get valuable insights into disease mechanisms.
So can you give an example of how Olink Reveal might be used in research?
Sure, imagine researchers are studying a particular type of cancer. With Olink Reveal, they could analyze the levels of those specific proteins, those 1,000 proteins, in blood samples from cancer patients and compare them to samples from healthy individuals.
Yeah, and that could help them identify proteins that are either elevated or reduced in cancer patients.
Exactly. Potentially leading to new biomarkers for diagnosis or even targets for drug development.
That's incredible.
Yeah, Olink Reveal has the potential to really accelerate discoveries in a lot of different fields. Drug development, diagnostics, personalized medicine. It's a really exciting time to be in the field of proteomics.
Definitely. And it's not just about the experimental techniques. We also have some really exciting developments happening in computational biology and bioinformatics.
Yeah, researchers are developing these sophisticated computational tools to analyze the huge amounts of data that's being generated by genomics and proteomics research. These tools are essential for uncovering hidden patterns, making predictions, and understanding the complexity of biological systems.
And one of the articles we have mentions spatial transcriptomics, which is this technique that maps gene expression within tissues. Can you explain what that is and why it's so significant?
Sure. So imagine you're trying to understand, the hustle and bustle of a busy city. You wouldn't just look at the city as a whole. You'd want to know what's happening in each neighborhood, on each street.
Oh, okay.
And that's kind of what spatial transcriptomics allows us to do with tissues. It's like creating this detailed map of where different genes are being turned on or off within a tissue sample.
Oh, that's fascinating. So instead of looking at gene expression in like a blended smoothie, you can actually see all the individual fruits and vegetables, where they're located and how they're all contributing to the overall flavor.
That's a great way to put it. Spatial transcriptomics provides unprecedented insights into how tissues develop, how they function, and how these processes can go wrong in disease.
So it's really powerful.
Yeah.
Another article we have highlights the use of machine learning to predict disease prognosis. Can you talk more about how that's being applied in genomics and proteomics?
So machine learning algorithms can be trained on these massive data sets of patient information, including genetic data, protein levels, clinical outcomes. And then these algorithms can identify patterns and correlations that would be impossible for humans to detect. That allows them to predict, the likelihood of a patient developing a particular disease or responding to a specific treatment.
So machine learning is kind of like this super detective sifting through mountains of evidence.
Exactly. They can identify those subtle clues that we might otherwise miss. These computational tools are really revolutionizing how we analyze and interpret biological data, leading to new discoveries and insights that are already transforming healthcare.
It's truly an exciting time to be following these fields. We're seeing breakthroughs in experimental techniques, computational tools, and our overall understanding of these complex biological systems.
And these advancements, they aren't just limited to human health. They're also shedding light on some other fascinating areas, like the history of art.
Art in genomics. That's an unexpected connection. Can you give me an example?
Yeah, one of the articles we have explores how genomics research has actually helped us understand the origin of those beautiful striped tulips.
Oh, yeah.
The ones that were so popular in Dutch Golden Age painting.
Yeah, I've always admired those striped tulips. I never knew there was a scientific explanation behind them.
Well, it turns out those stripes, they're not a natural variation. They're actually caused by a virus.
Yeah.
The tulip-breaking virus, it interferes with pigment production in the tulip petals. And genomics research has helped us uncover the genetic mechanisms behind it.
Wow. So it gives us a new appreciation for both the artistry of nature and the power of science. Who knew a virus could be responsible for creating such beautiful works of art? So from the intricacies of DNA folding to the complexities of the gut microbiome to the surprising connection between viruses and art, we've covered a lot of ground today. What do you think is the most significant takeaway from all of this research?
For me, it's the convergence of different disciplines. We're seeing biology, chemistry, computer science, even art, all coming together to drive innovation and discovery. It's really a testament to the interconnectedness of knowledge and the power of these collaborations between different fields.
I completely agree. So inspiring to think about all the possibilities that are out there as these fields continue to evolve and intersect. What questions does all of this research spark for you?
Well, it makes me wonder how these advancements will shape the future of healthcare. Like, will we see personalized medicine become the norm? Will we be able to cure diseases that we currently consider incurable? Will we unlock the secrets of aging and extend human lifespan? Those are just a few of the many questions that I'm really excited to see answered in the years to come.
It's truly an exhilarating time to be following these fields. We're witnessing a scientific revolution, and it's transforming our understanding of life itself.
And it's a revolution that has the potential to improve the lives of countless people all over the world.
Well, that's about all the time we have for today. Thanks for joining us on this deep dive into the fascinating world of science. And keep that curiosity burning. We'll catch you next time.